Well, good morning, everyone. Before I start, I was thinking back at what Steve said, and I really want to echo the leadership that Paige has shown. And what's remarkable is that she says things in such an unassuming way that where she, I think, really deserves an incredible amount of credit for pushing this field along, and I really applaud her ability to do so and to be persistent despite some of the challenges that I think we all faced in the past. So in this talk, I'll cover some of the metabolomics data that we generated as a part of this consortium and as a part of this network. I don't have any conflicts of interest related to this content. So very briefly, I'll talk about some of the metabolic profiling that we did because we really knew nothing about it, and I'll talk about the reasons why we went into this briefly as well. And also, how this kind of an analysis, it's not just a one-and-done study. It really feeds off a number of other hypotheses that continue to evolve and may very well have therapeutic implications in a variety of ways, and I'll show you a couple of examples around that as well. So part of the motivation to run a metabolic analysis really stemmed from a number of studies that we had been doing over time. In the past, when we first started working in this field, a lot of the work was really because of specific hypotheses around genes or pathways. But as the technologies started to evolve, we really wanted to take unbiased and global approaches to understand what pathways are activated in response to some of these perturbations. So one of such experiments to try to look at specific cell types of interest that we did was to do a 2D proteomic analysis of cells that had been treated with norepinephrine at stress-relevant levels. That always, generates quite a bit of data, and we picked out a lot of spots that were differentially either the protein levels changed or the phosphoprotein levels changed, and then created a network from this kind of unbiased analysis. SARC ended up being a dominant node that we pursued in this particular paper and showed how stress hormones can activate SARC in a very unique pathway, and I'll be happy to discuss that more in detail a bit later. But the reason I show this figure is that a number of metabolism-related genes kept coming up. Moreover, from some of the unbiased analyses or arrays that Steve had run, same thing, that there were pro-inflammatory pathways and some of the metabolite pathways that we had clues related to, but didn't have specific data around. Now, metabolism, again, this is a common slide that many of the metabolism speakers show, but just as a background, it's important to understand that a lot of this field started because of the interest in aerobic glycolysis as a source of fuel for rapidly dividing cancer cells. It was a few years ago, it was accepted as one of the hallmarks for cancer, and the definitive evidence for whether it's a true driver or a byproduct of evolving tumor biology, I think, continues to evolve in terms of its understanding. But regardless, I think that at this point, the ability for tumor cells to develop aerobic glycolysis is fairly widely accepted. To try to understand, both in the cancer versus normal context and to what extent then biobehavioral pathways can overlay on metabolism, we took a set of samples that Susan had collected over a period of time, and these are samples from patients with high-grade serous ovarian cancers. And then for this particular initial discovery work, we used around 15 normal ovarian tissues. Since then, for validation purposes, we've also used fallopian tube samples. As some of you may know, about 40% of high-grade serous samples tumors start from the distal end of the fallopian tube and not just from the ovary itself. From this kind of a analysis, there were around 300 some metabolites that were different between the two groups. And 170 some metabolites were significantly altered in high-grade serous ovarian cancers. 140 or so were upregulated and 30 were downregulated, and this is just a heat map showing the alterations in these metabolites. Not surprisingly, there were a number of glycolytic related metabolites that were elevated in cancer, and then also pentose phosphate pathway metabolites. There were also long-chain fatty acid metabolites and a number of other pathways that we've recently reported that were altered in cancer. And inflammation-related metabolites, again, fairly significantly elevated prostaglandins and related metabolites in tumors, and some of that's not a surprise. We know that there are inflammation-related metabolites that are altered in cancers. The power of this group of samples was that Susan had done a fairly extensive set of biobehavioral profiles on these patients from whom the samples were derived. Now, there are a lot of ways to look at this data. We for some of the analyses, looked at high versus low depressive scores, because some of these scores really seem to be consistently related to patient outcomes, not just in ovarian, but also Lorenzo's reported in renal cancer and in other cancer types as well. And indeed, a number of these inflammation-related pathways, especially the prostaglandins, were indeed elevated in those with high depressive scores. Anytime that we now do a lot of the projects, we use sort of an iterative process between human samples, mechanisms, as well as mouse model systems. Steve alluded to some of the mouse model systems that we've used, and for stress-related work, a lot of the systems that we use relate to daily restraint stress. Very similar metabolic profiles in tumors from animals that were exposed to this kind of a restraint stressor, where you see elevations in the same kind of prostaglandin pathways that we saw in the human samples as well. With that as background, we started to think about, what might be some of the mechanisms by which these kinds of alterations may come about. So PTGS2 and PTGES are the two synthetic enzymes that are in the prostaglandin synthetic pathway. There's one catabolic enzyme in this particular pathway. If you expose cancer cells that contain beta-adrenergic receptors, you see significant elevations in both of these biosynthetic enzymes, which would then relate, obviously, to production of the relevant prostaglandins downstream. Even if you look at a protein level in response to norepinephrine, you see significant elevations in PTGS2. Both of these, by the way, are inducible enzymes. They are, so that makes sense that in response to specific stimuli, they would indeed change in terms of their levels. If you look more specifically at a variety of agonists as well as antagonists, what we find is that it's really the beta-2 dominant agonists that seem to drive elevations in these synthetic enzymes, and that if you use a set of broad beta antagonists, such as propranolol, you can largely abrogate the increases that you see in both of these synthetic enzymes. Moreover, at a mechanistic level, we try to search for what might be really mechanistically driving the changes in these inducible enzymes. So we did a promoter screen and found that NF-kappa B contains binding sites in both promoters for PTGS2 and PTGES. And this is a chip analysis basically showing you that indeed there's a much greater binding of NF-kappa B to the promoter sites for both of these inducible enzymes, and the P65 and P50 levels in response to norepinephrine indeed go up significantly. Now, how do you perturb these kind of pathways then at a mechanistic level, but at a biological level? So one of the areas that my group focuses on is we develop nanoparticle systems for a systemic delivery of siRNA. One of the first systems that we've developed is something called dioleo phosphatidylcholine, or DOPC. This is a neutral nanoliposome. It's a multilamellar carrier. It's got an average size of 65 nanometers, so it's indeed in a nanoscale. We've done a large number of studies over the last 10+ years to demonstrate. So the red color here is the fluorescence for siRNA, that indeed it gets beautifully into the tumor microenvironment. And the green is a stain for scavenging macrophages. It really doesn't get taken up by macrophages because it's neutral in charge. This particular carrier is in phase 1 clinical trials at MD Anderson. We've brought all the way from ground zero into now phase 1 testing. And in leukemia patients, it's already in phase 2 testing for an antisense oligo. Using this kind of an approach, we can systemically target a given pathway of interest. And in this setting, if you look at these orthotopic mouse models of ovarian cancer, systemic delivery of siRNA, in control siRNA, you see significant increase in tumor growth as well as the number of metastatic sites. But if you block PTGS2, you not only block the elevations in the inflammatory metabolites, but you completely abrogate the increases in response to that kind of a daily restraint stressor. This is, and if you look at a tumor level in response to stress, again, you see elevations in PTGS2 levels, which are abrogated if you use that kind of a systemic delivery modality. This was a very direct orthotopic model because we surgically implanted tumor cells directly into the ovary, so this is not an IP injection. So you can indeed look at metastatic sites as well, and what you note is that where we silence PTGS2, you really substantially reduce the metastatic patterns in these kind of mouse models. Now, there were a number of other metabolites that were elevated, and one that caught our eye that very briefly, I just want to show you a little bit of data around in the full metabolic profile, by the way, recently we reported as well, was a neuronal metabolite called NAA, which was a surprise. We certainly didn't expect to see that, but it was massively elevated in these tumors. Now, as you know, most mass spec-based modalities don't give you a quantitative answer. You can look at relative changes, which is indeed what we in our discovery set looked at, where in the tumors, again, there's pretty significant elevation. So we developed NMR-based modalities for getting a quantitative assessment of several metabolites, but here, specifically the NAA levels, and again, pretty profoundly elevated in these tumors. NAA has been reported, it's a metabolite actually that's now known to play a role in Canavan's disease. So it does cause other disease problems as well. But in an ovary setting, we certainly didn't expect to see a neuronal metabolite to this extent. There are a couple of small studies now that have reported pretty high levels in cyst fluid from serous tumors, and it may be a potential biomarker as well. And some groups are also reporting it in prostate and breast cancers as well. Biological roles before this work, largely unknown in the context of cancer. Now, again, overlaying informatics can be really quite powerful. We work with Keith Baggerly, who's a forensic bioinformatician, very powerful tools that he's created. This is a pan-cancer analysis using all the TCGA sets for NATSL, which is a biosynthetic enzyme for NAA. What you note, the pink here is, sorry, let me go back, is ovarian cancers. So what you note is that if you look across, 20-some different tumor types, so each of these tumor types has about 500-some samples, so this is a large amount of data. Ovarian cancers indeed have fairly high levels of the synthetic enzyme, as do melanomas and select other tumor types. So you can be smart about what tumor types you really want to go after if you want to pursue this kind of a metabolite. So if you perturb this pathway and you do reverse phase protein analyses to look at downstream pathways, what we find is that things such as the AMP kinase pathway go up in response to silencing or shutting down NAA, and you see downregulation of a number of cell cycle related proteins. Again, at an in vivo level, we use our DOPC platforms. If you silence NATSL to reduce NAA levels, fairly profound reductions in tumor growth. We've done similar experiments in melanoma because of the TCGA data that we found. And if you combine it with certain cytotoxic drugs, really robust anti-tumor activity in this context. So in summary, global metabolite levels in the context of this network were really allowed us to do a first analysis of human tumors where we had well-related biobehavioral profiles, and then we had an additional benefit that Steve had run some arrays on these, and now we're relating the genomic profiles with metabolite levels, which is the kind of prosomics analyses that we really want to be able to do. Mechanistically, some of the stress pathways indeed result in elevations of these pro-inflammatory metabolites, and shutting down these pathways reduces metastases in vivo. And I didn't show you the data related to patient outcomes and so on that we also reported in that paper. I want to acknowledge the work not just from my lab members who carried out this work, but especially collaborators such as Susan, Gabe Lopez, Steve Cole, and the support from the network as well. Thank you very much, and I'll be happy to take questions.